Check, check.
Yeah, get her presentation up. All righty, everyone. Hello and welcome, Reef Fest 2024. How about this? We are really honored to have everybody here. It's an exciting day. It's an exciting time at Reef. If you've been around, if you were here at our open house last night, you might have seen the new building that's springing up on the campus, at the Reef campus. We have an amazing staff that we'll bring up later and share with you. But before we get started on our amazing speaker lineup, I just wanted to say a few thank yous. We have an amazing crew here. This is a perfect space to do our seminars. It's the government center here provides us so much support and allows us to kind of crash their working environment. We've got our amazing David Hartman up there out in the booth. Yes, he's putting his hand out. Thank you, David. And Daryl Duda, his right-hand man down here with giving the mic checks. And Stacy, our own Stacy and Sierra up in the booth. They're going to make all this possible. And the amazing thing is not just for all of us in this room, but we're live streaming this out on Facebook. So all of our folks, friends, family, members all over the world could potentially be watching right now. And it will also be archived on our YouTube page. So that was an advance we made a couple years ago that's really been great because we do these great speaker lineups for Reef Fest. And then it was just for the lucky few who was in this room. So we're really happy to be able to archive them now on our speaker page. We want to do a big shout out to our title sponsor, First Horizon Bank. You guys, I know there's been a lot of bummer about not getting out on the water, but our eco excursion hosts are always very helpful to help get us all arranged, and they were all ready to have you all out on the water. So I'm sorry that the weather hasn't quite cooperated, but hopefully, fingers crossed, tomorrow it's going to be good. If you didn't get a chance to try the coffee in the lobby, Caldi's Coffee, she's amazing. She'll be at all of our events and really good coffee. So if you're a coffee fan, she'll be out there at the break when we break for our happy hour. Definitely check her out. So our first speaker is, I'm so happy. It's been really fun to get to know Natalia the last 24 hours, basically, but we've just really connected. She comes to us from the University of South Florida. She's a Ph.D. candidate there, and she's a born-bred Puerto Rican, and just has some amazing stories to tell about coming up and pursuing her craft of science, but also a deep connection of helping bring science to the masses and science communication, and helping mentor younger individuals, whether that's college age kids or younger. And as you all know, that's a deep connection that Reef has that's at the core of our mission. And so we're really excited to learn and hear from Natalia. So she's going to share with us, let's see, her title, Upside Down Jellyfish as Bioindicators. She's going to share with us now. So thank you. Thank you.
Good afternoon, everyone. I am honored to be opening this seminar series. I told Christy yesterday that I've been aware of Reef since I started diving in 2012, and I never thought that I would be here speaking to you all today, and I'm very excited to talk to you about the work that I've been doing in the past 4 years as a Noah Margaret Davidson Fellow and at the College of Marine Science. So let's get into it. So I wanted to start with, if this allows it. Yep. I'm trying. We're live, everybody. There we go. Let me see. All right. So to start, I wanted to, go back to basics. What are bioindicators? And we've been using bioindicators since the 1990s, which was the other day. I was born in 1990, so this is very, very recent. So bioindicators are these type of organisms that exhibit some type of change in their biology, in their behavior, due to an effect of change in their environment, whether it's physics, chemistry, or just anything that is not what they're used to, right? And we've been using algae, invertebrates, plankton, jellies. I'm gonna use jellyfish and jellies interchangeably. I know they're not fish, so I'm just giving that heads up. So in the top portion here, we see Ulva lactuca, which is the sea lettuce. And this is usually a bioindicator of just fecal coliform or just human waste. And then we have in the bottom cannonball jellies, which in this particular scenario is not that there was a chemical change, but the people that were trying to get rid of them didn't know that when you slice them, they release their gametes and they just made the problem 10 times bigger. And this is what we call human impact. So why do we want to study jellies? Well, they respond quickly to anthropogenic activity like overfishing, changes in pH, and even though that there is a common knowledge of like, oh yeah, jellies are these robust organisms that can withstand very harsh conditions. This is not true for all jellies. And most of the information we have about jellies has to do with pelagic, so the open ocean. But we don't really talk about the coastal areas, the ones that are impacted by ourselves. You know, we all love the beach, or if you have a beachfront house, or in general, if you enjoy beach activities, you are being exposed to the coastal zone. And so the blooming conditions of these organisms is still unclear. And I've had a professor ask me a couple times, so why are they upside down? Why are they upside down jellyfish? Is it because the ocean is warming? Is it because it's changing? No, it's their behavior. So a typical jelly would look like the bell is swimming upside. The upside down jelly has a behavior that the bell acts like a suction cup and it adheres to the substrate. So it's semi-sessile. They can't swim. They just, they're lazy. They choose not to. Why would they move? Because they are actually, like coral, symbiont bearing. And they expose their oral arms, which what you would see the tentacles, because that's where the symbionts are mostly concentrated. So they receive most or all of the metabolic requirement from those symbionts, but they are able to feed heterotrophically, which means that they can consume plankton. They are females and males. They can reproduce sexually and asexually. And who here has been personally victimized by the contactless sting of upside down jellies? Yes, so this actually has a name. It's called the casiosomes, and they can sting you without you even moving them. So, as I said, they are very similar to coral, and because they're not protected like coral, they're usually used as a good model organism to study symbiosis, but much more than that. Cassiopeia is a model organism for fluid dynamics, for venom studies, development, and even just like developmental biology. So it's a very versatile organism. But what about their bioindicator capacity? So this has been a rising topic that has started in the Bahamas and has migrated to Brazil and even in Australia. And so they're the only known benthic jellyfish. They're pretty easy to spot. They don't really move much. So everybody can basically have a general idea and identify, that's a Cassiopeia. So they're distributed all around the globe in the tropics, and they're preeminent in human impacted areas. So in Australia, they've performed some experiments where they've seen that they respond negatively to herbicides. They also respond negatively by the bioaccumulation of trace metals. So they're kind of like also absorbing what's in the water. And dredging for aquaculture facilities in mangrove areas promotes the space for them to invade, because as you're removing all these things, the sediment is resuspending. You have all those recycled nutrients that their symbionts can consume. And they thrive there because there's no competition, right? It's a new open space. And then finally, there was a study recently in 2021 about them being a microplastic sink. They sampled Jupiter Beach, the Florida Keys, and Sarasota Bay, and they found the higher concentration of microplastics here in the Florida Keys. So I invite you to see that article. Is really interesting. But the other thing that in my anecdotal and in this research that I've been doing, what I've noticed is that they can also potentially indicate latitudinal expansion. So the warming temperatures are actually because they're in the tropics, we found them usually in the Caribbean or in Indonesia, but now we have found them all the way to St. Pete Beach. And in this picture on my left, now you're right, it's in a residential dock. We found Cassiopeia that was completely bleached. They've never been reported there, and that is as farther north as we've seen them. So with all this information that we've seen, that they're widespread and they can potentially be used as, bioindicators to our service, that's where my dissertation wants to answer. So can they be used as bioindicators of anthropogenic nutrient enrichment? And so to answer this question, I have three main objectives. The first one was going to the literature and do a review using the papers to see what have we done and where should we go next. The second one, I wanted to determine the substrate of preference for the polyps. So it's a developmental stage that influences the settlement in the field. And this is particularly because we want to know about their blooming conditions. What makes them bloom? Are they more susceptible to bloom next to anthropogenic areas? And then finally, I wanted to assess the distribution of Cassiopeia in different degrees of human impact in high and low human populations. So for now, I'm going to talk to you about the objective 2, but I'll give you a preview of the article that's going to be published at the end of this month in the Bulletin of Marine Science. There's a QR code if you want to see the article. But essentially, in my literature review, what I found is that 73% of nearly 200 articles and theses and dissertations, 73% have been lab-based experiments. That means that only 14% has been just in the field. So there's a lot that we need to learn about their field ecology in general. We also see that symbiosis and their life history is one of them, is the main topic of interest and the most researched topic. And bioaccumulation or bioindicator ecology really is not as abundant as we want to. So that's the reason why I wanted to do this research. So what we know about the Cassiopeia life cycle is that they release a little larvae that doesn't have symbionts, right? And again, these are all the life stages. So this is a polyp, and the polyp needs to have enough concentration of ammonium in the water, and there needs to be symbionts, and the water temperature has to be higher than 20 degrees Celsius for the polyp to develop these tentacles, ingest those symbionts, and then release the little jellies that we call Ephyra. Now, this sounds all great and dandy, but all we know about their life history has been in controlled environments. So does this really translate to the field? With all of these requirements, how do they form these massive blooms, right? So my story takes place in Hobos Bay National Estuarine Research Reserve in Puerto Rico. And Hobos Bay is a great area to study bioindicators in general because it has a history of long-standing human impact. It was the location of the largest and most productive sugar mill in Puerto Rico, and it ceased operations in 1980, and then the year after, it became a sanctuary. And what makes Hobos Bay a different estuary is that it's a coastal plain estuary, meaning that after the last glacial period, lowlands and river valleys were drowned, meaning that groundwater is the main water source of this of fresh water in this particular estuary. And so I wanted to kind of do like a study where it was in a controlled environment that was human impacted and had been promoted potentially for Cassiopeia. So after the sugar mill closed, a lot of these terrains of sugarcane crops were abandoned around the island. And after tropical storms, they would get flooded by either high tide or the rainfall. But managers noticed that birds would often forage and use this area as habitat. And so in 2019, they decided to dredge this area and make it a lagoon to promote the bird, the shorebird populations in this area. But of course, who do you think liked this the most? This is an aerial picture of Cassiopeia that invaded the lagoon. All that yellow that you see, those are all jellies. And they just appeared then within a month of dredging. And so that's when they called me and they said, well, you like jellyfish, what's going on here? So I went there and I wanted to do like my Pinterest image of what I wanted to do and see how they settle. is take what the literature said that promotes that settlement, that promotes those blooms, and kind of build a trap to see if I can lure them in and see what they prefer. So my idea was having all this mangrove leaves, seagrass beds, the seagrass leaves, and then some fertilizer pellets that when they release those larvae, they would be attracted to these settlement tiles. And so in that manner, we would see if the polyps had set on the tile or on the material itself. So we did this study for 6 weeks. And what we noticed is that we did not see any polyps, but we did see a lot of other things, because you open Pandora's box when you're studying anything in nature. The more you find out, the more questions you have. And so we found juveniles that were settling on the vegetation material. We also found that the adults would be getting closer and closer each week to those materials that, like the red mangrove leaves and the seagrasses. But we also saw a potential new predator, which were sea hares that were biting off the oral arms of these organisms. And there's not a lot of predators out there for them. So again, anecdotal information is just as valuable as any statistical analysis that you do. So after an event of king tides and storms, the population had a major die-off, and now they have a bunch of egrets and shorebirds. So now the pond is doing its thing that it was meant to do, but no more jellies in that aspect. So we always learn from our mistakes, right? So the experimental design definitely needed to change. We used rebar. Rebar is made of iron. Iron is a very limited element in the ocean. So when it's available, just everybody goes ham. But we wanted to change to also have a more open trap. And so this is what led us to do the case study. In the case study, we designed 3 traps that were a little bit more free, with the same concept of having those settlement materials that we found in the literature. And then we wanted to do it in sites that were Cassiopeia were present, and we wanted to do it in triplicates. That's the good thing for statistics. Always keep it in 3. So we did keep the monitoring to 6 weeks because of the availability of the resources, such as the personnel and the boats. And we had a similar approach, as I mentioned, and we also monitored the settlement closely. So the first idea that we had was doing 2 acrylic plates, right? One on top of each other, tied to a PVC tube. And we deployed this in the lower keys. And the idea was to use 3 types of degradation stages of the red mangrove leaves. My grandfather helped me to build all of this. He's retired, thankfully. He's 90 years old, but he's an innovator. And so we designed a second plate that was an acrylic plate, but then a mesh bottom. So that wasn't as heavy as the first acrylic plate, which we always had to dig up because it was so heavy, and everything was muddy as well in all sites. And so the different approach here is that I took a female and I took her little eggs, and then I kind of put the eggs in those little holes when we were in the water to see if that would promote settlement. Now, my grandfather said, I don't think so. I think you have more here to work with. And I said, okay, so what do you suggest? He said, let's do like the telephone cable. You remember the cans with the rope? So basically, this is a telephone toy, as he mentioned. And we used 3 different settlement substrates. So essentially, all of them were from the same site. None of them were picked or brought from another site. We wanted everything to be kept from the same site. And we did this in triplicates, and we just put holes in these bottles and then tied them up in like a little cable very nicely. Now, number 3 didn't work. Who says number 1 worked? Show of hands. Who says number 2? Those who bet on number 1, you were right. My grandfather's design was the one that won. And we found 25 polyps settled in the most gross, filled biofilm, filled degraded leaf that you could ever find. That's where they were the happiest. There were many. They were very chubby. Oh, they were very happy. And this is a major leap for us in the Cassiopeia world, because now we don't have to go Easter egg hunting. We can just trap them and start doing life history experiments, but with life organisms from right there. So I'm very excited about this. And we actually have a provisional patent from Arizona State University and SkySong Innovations for this great experiment that we did as a family. And so now I want to wrap it up with the assessment of distributions. Now, this is the chapter that I'm currently working on the data, but I wanted to give you a teaser of what we found. So in this chapter, I had three main objectives: monitor, test, and compare. I wanted to monitor these populations in the different degrees of human impact, test different sampling measures that managers and people in the field could use in a time and be time effective, and also compare the physical and chemical conditions from the areas in which they bloom to see if there was any similarities. And so again, this took place in Hobos Bay. And for high human impact, what we decided was 1 kilometer square, in a radius of 1 kilometer square, the amount of buildings within that radius. And because Puerto Rico is highly populated and everybody constructs anywhere because the coastline is inherited by other people, we decided and determined that this would be like the high impact area because you have a lot of communities that build in the area in the watershed. And then we have mild human impact because even though we have a little bit of a construction down here, there's a lot of agricultural activity up in the watershed. And then the lower keys, because of the circulation pattern of how the water moves, we determined that that would be the low human impact because it's not as influenced by the runoff. And so as we go in the site, we collected the water quality using a multi-parameter sonde where we took the pH, dissolved oxygen, temperature, salinity. And the first method that we used was a transect. So we put a tape measure that was 10 meters long, and we established 5 of these. And what we had was a mount with 2 cameras. One would take around 10 frames per every 10 seconds, and then the other one would be just recording, so we can compare them along the transect. The second one was the random quadrat toss, because we want to estimate what's the % coverage of Cassiopeia in the field. So just as it sounds, we have a PVC square, basically, of 1 meter squared, and we toss it around 10 times, and then we estimate what's the percentage of Cassiopeia versus seagrass versus sea urchins that we find in the area. And in addition to doing all that fun picture and field work, we also collected DNA samples, so I got to chop up some jellies. We also collected seagrasses and water to test the nutrients. Now, why both? Water gives you a snapshot of how things were right there. Water keeps moving and circulating. But seagrasses and algae have a long-term accumulation of the nutrients. So you have a good comparison of what happens short-term and long-term. And finally, we also collected sediment grain size analysis, sediments for grain size analysis and foraminifera, which are these tiny protists that actually have already been established as some bioindicators or stress tolerant. So they're kind of like our tutors. So if we find more or less the same forams that we find in these high or low human impacted areas, and we see Cassiopeia, then that might tell us a little bit more information of our inferences as a bioindicator. But focusing on the DNA part, which is the one that I was focusing on in this aspect. So think that you're in the supermarket, right? And every item has a barcode. So just like the items, we also have a barcode in our genes. So the genes that I targeted are specifically for Cassiopeia. So when I did all my extraction and the analysis, essentially, it's just like going to the supermarket, and it would give me my species with comparison to the database. And why do we care about this? Well, the same species can look 4 different ways, 5 different ways. They have a high, what we call phenotypic plasticity. They can look very different ways, the same species. So we wanted to know if the areas that had high human impact or low human impact would have the same species or not. So there was no significant difference between high and low or high and mild. So at the end of the day, after a statistical analysis, we determined that it was only high and low human impact. When it came to the abundance of Cassiopeia, it's interesting because even though we did see some Cassiopeia in the bottom, in the keys, we did see that it varied very largely with Halophila. It seems like there's a battle of the benthos. This invasive seagrass is kind of displacing and competing for the space in where they are inhabiting. And you can clearly see that as we see the percentage coverage. In here, in this site, you can see how as Cassiopeia is decreasing, you see how that halophila is increasing. When it comes to the other, sorry, that was the mangroves. This is the Lower Keys. We did see one pulse of the Cassiopeia blooms, but then after that, they just declined completely, and then the halophila declined completely. We don't know what happened there, but right now it's a dead zone. But the other two sites that were control sites, we did not see any Cassiopeia, and we did see a more diverse amount of macrophytes in that area. Now, in this very busy plot, this is basically all of the parameters that we looked at, and what I want you to take home from this is that we saw a negative correlation between nitrate and salinity. And what does that mean? One of the things that it can mean is human impact, so human waste or some type of agricultural waste, and that would make sense because in the watershed on the top, as you can see, there is some agricultural activity. So further information will be processed as I do the research, as I need to finish next semester. So stay tuned for that. And finally, we also see more ecological interactions. As I said in the beginning, there's not a lot of work with the field ecology of these jellies. So I assembled a team of bright scientists that are working on the different aspects, because as you saw, I'm collecting a lot of stuff and I'm only one person. I can't really do all of it, but it actually benefited other students to create their own projects. So we found that there is pretty much a similar species, Cassiopeia samacana, which we find it also here in the Keys. And the phenotypic plasticity is very wide. We saw this foram called ammonia. That's a stress-tolerant foraminifer that was very abundant in the high human impacted areas, which makes sense. And then potential new predators include barracudas. We don't really have a big video about this, but we did see, wherever you see Cassiopeia, you see barracudas, and they're missing some limbs, so you do the math. We did see some butterfly fish nipping at them and some parchment tube worms, and finally, the seagrass that is just dominating the Caribbean right now, Halophila, shows an inverse relationship with the Cassiopeia abundance. And in a nutshell, exploring this potential of bioindicators is increasingly important and is of recent interest because of all the changes that our oceans are withstanding at the moment. So we do need this type of tool to let us know how are the conditions. But what we've seen so far is that Cassiopeia may be just more than just a bioindicator. It might be providing ecosystem services as well. And finally, this work at Hobos Bay was a baseline in Puerto Rico, but as things are changing, this can be applicable not only to the Caribbean, but in the future, it might be used here in Florida. And if you want to know a little bit more about what I did, there's a Nature Tools of Trade article. There's the QR code there. I'd like to thank the Noah Margaret Davison Fellowship for funding this project, as well as all these entities, the McKnight Doctoral Fellowship for funding my travel very quickly one day to another to come here to Reef Fest. I want to thank Christy and the Reef Fest people because this is a great opportunity. It's an honor to be here. I'm humbled to share my research with you all. And I couldn't have done it without my family, my friends, my pets, the REU program at USF, and the Hobos Bay personnel. And if I've convinced you that you like Cassiopeia and you want to study Cassiopeia or learn more about it, you're in luck because we are planning our 8th annual International Cassiopeia Workshop right here in Florida Keys. So the symposium is growing, and the symposium area is going to be in the Whitney Laboratories, but the workshop aspect where we collect data and we do experiments is going to be right down the road here in the FID Marine Lab in Key Largo. And with that, if you have any more questions or want to know more about me or contact me for anything related to jellies, that's my information right there. And I thank you all for your time. Muchas gracias.
Wow, that was amazing. Okay, we have questions in the audience. We've got folks. So Carolyn, I think it looks like Jack's got a question right back there. So if you've got a question, just be patient, wait for the microphone so our folks online can hear the question.
Wonderful talk. Thank you very much. You used a term for an organism that consumes plankton. Could you repeat that word for us?
Yes, planktivore.
Oh, planktivore.
Yes.
That's easy enough.
Yes.
So then is planktonivorous a word? For example, carnivore there are carnivorous organisms, herbivorous organisms. Oh, do you think that Cassiopeia is a planktonivorous?
Yes, absolutely.
Thank you.
Now, I won't say, I don't have a degree in grammar, but I agree with your, I agree with the association, so yes.
I don't know much about jellyfish, but I'm very interested in fish, real fish,
Yes.
that eat plankton.
Yes.
And I have not seen the word planktonivorous in the literature, but I'm going to use it.
Yes, I agree. We should all start using it.
And if I get any problem, I'm going to talk to you.
Yeah. Scan the QR code.
Thank you.
Is that the only species that turns upside down and eats?
For now, it's not the only species of cnidarian that turns upside down. There is another hydrozoan, so related, that is not upside down, but it lives in the benthos. But so far, this is the only upside down jelly that exists.
And that's their only way they feed.
Well, they have symbionts, so they.
I mean, they don't swim.
They swim. It's just they also have a sleep-like state, which is very interesting. They just had them in fish tanks, and they had grad students poke them to see every so often if they wake up or not. But they do swim. But there's so much to be done in the field. We thought about putting some cameras to see what's their behavior at night. But they do swim. We've seen them swimming, especially when I pick them up, cut their limbs, and throw them back. They swim back down. All for the good of science. They grow them back. It's okay.
So do they get any of their energy from photosynthesis? And if so, how does the impact of maybe sediments from the agricultural activity that you were describing affect that?
That's a great question. Yes. So their symbionts with photosynthesis provide their metabolic requirement. And if they're close to areas where there's a lot of turbidity, they're not going to be there. It really causes them a lot of stress, and they just move to areas that are low movement, more pristine. And so that's why I mentioned that my advisor, Dr. Pamela Halleck, she has the idea that it's more than just a bioindicator. It also indicates that there's good water quality as well for what we've observed. But since we're working still with the data and the nutrients, we don't want to discard the fact that they could be bioindicators. But turbidity definitely affects their performance. They can bleach like coral as well. That's another thing that we have been finding in the field. So yes, anthropogenic activity and all that sediment runoff after a big storm event, they wipe out.
Follow-up question to that question is the symbiont photosynthesis metabolistic behavior the only way they're getting any nutrients, or do they also hunt and sting like other jellies?
Well, that's another great question. And the answer that I have for that is that we are studying the casiosomes, which is the contactless thing. They release this gelatinous kind of cloud, and it does have symbionts, it has stinging cells, it has amoebocytes, but we don't really know what the function is yet. So they could be casting a net to feed from like plankton or fish eggs or whatever's around. But they definitely have multiple ways to get their energy, whether it's symbiosis or just by filter feeding.
Thank you.
No problem.
Is you say Cassiopeia or Cassiopeia?
Both.
Either. Is it native to the Keys?
So the phylogeny is tracked back to Indonesia, so the Southwest Pacific. But it originally here, it was introduced by we don't know what. We don't know how. Maybe ballast waters or even in the aquarium trade. But they have been in the Keys naturalized for years now.
Are there different species in the Keys? Because, for example, the ones at Founders Park you can pick up and they don't sting, and the ones at Penny Camp sting. Is that not that I would pick them up?
I mean, you can. They're not going to really complain, but you're probably going to get stung. I have not encountered a Cassiopeia that didn't sting. That would have made my field work a lot easier. But there are different species in the Keys, and there's a lot of studies. Actually, most of the studies in Cassiopeia have been performed here at the Florida Keys, specifically in Key Largo.
I was wondering if you are using citizen scientists for the study of the latitudinal expansion of the range.
Yes, actually, I'm part of the Cassiopeia-based network, and the map that I showed you was made by the student Miles from Texas A&M. It's from iNaturalist. So if you use iNaturalist and you're taking pictures and uploading them to your app, you're contributing to all of this data set. And I was talking to Christy about this. The boat operator, Luis Ortiz, he's a native person from Guayama, Salinas area, and he was crucial, I would say key, to finding all of these sites because he grew up there and he knows all the anglers, and the anglers would even tell you information about, well, after a storm event, we don't see them anymore, but then a couple months pass and then we see them again. So the citizen science for me is the most valuable information we can acquire, because as scientists, we're not there all the time, but these people really depend on the resource every day. So what better way to find out about any patterns in the ecosystem than using citizen science? So yes.
Wow, everybody loves jellyfish. See, you thought it was a fish crowd.
Welcome to the world of jellies.
Yeah. Okay. So that was really great. Thank you so much, Natalia. We're going to get Kramer Wimberley set up for our next talk. So you could come on up, Kramer, and we'll get you sorted with the microphone. And while they're doing that, just a couple things. I wanted to give you some logistics. So we've got, if you didn't find the restrooms already, there's restrooms in the lobby where you first came in. There's our silent auction, and our little pop-up store is here in the lobby, or in the kind of the hallway that you walked through. After, when we're done and we break for the happy hour, all of that will move out into the lobby. So it'll be a lot easier to see. If you didn't get a chance to check out the silent auction or the pop-up store yet, it will be out there later. We're going to hear in a couple minutes from Kramer, and he's going to share some really inspiring work that he's been doing for a very long career, almost 40 years. He's been working with the scuba diving community to help engage and transform lives in much the same way that Reef has done. And so it's a really inspiring story that he's going to share. And then after Kramer, that's when we're going to do our little memorial and remembering Paul Human, co-founder. And then after that, we'll break for our little happy hour social in the hallway, not the hallway. We'll all gather in the hallway. No, we're going to go out into the lobby at that time and kind of celebrate, and then we'll come back in here to hear Rob Robbins and his amazing work on fishes. So that'll be how the rest of the evening goes. We're not going to take a break. So if you do need to do a quick restroom run, we're going to take the break at the happy hour, but not until then. So just feel free if you need to do a quick bathroom run, a quick stand up, but that's how we're going to do it. So we really appreciate everybody being here, and freezing. It's freezing in here, isn't it? So I'm sorry about that. Okay, so let's see. Do you have your? Yep. All right.
Are we waiting for the bathroom run? Are we waiting for the bathroom run? Or
no,
yeah,
we're good. You can't wait. They'll just, it's a rotating door. Okay, so like I was saying, Kramer has had a very long career working with in the diving industry. He's a longtime diving professional, almost 40 years. That's an amazing career, given that there hasn't been scuba diving for that much longer. And as he's going to share the story of this amazing organization, Diving with a Purpose, and some of the great work that they've done, both to look for cultural heritage resources, but also engaging divers as citizen scientists to document reef health.
Yes.
So, yes. All right. So take it away.
Thank you, Christy. Apologies for the seat. I'm a little taken aback. I've got hip surgery scheduled in a couple of days, but I did not want to miss the opportunity to come down here, and I already committed to working with Reef and supporting them in their efforts there. You're actually a part of our citizen science program with our youth component. Good morning, everyone. Thank you for inviting us here. My name is Kramer Wimberly, as you already know. I am a board member with an organization called Diving with a Purpose. Diving with a Purpose is a citizen science-based organization. Started about almost 25 years ago. Right? Diving with a Purpose actually has two branches right now. Right? When it was founded as an organization that worked on documenting historic shipwrecks, both in the national park and in the National Marine Sanctuary. I am going to briefly go through Diving with a Purpose, Maritime Survey Archaeology, and then get into the love of my life, absent my wife, Diving with a Purpose, collective approach to restoring our ecosystem. All right. And the emphasis throughout the majority of this presentation is going to be on the first letter and the first word, which is collective, right? Because everywhere I go, whomever it is that I'm talking with, once we begin talking, we're family, right? We are a family of people who are passionate about the ocean and attempting to restore the ocean. The vast majority of people in this room are also divers. So to have an opportunity to speak to divers about the environment with which we derive most of our joy, right, is additionally enjoyable for me. Right? I have a challenge with following slides and presentations because I get excited, right, about whatever it is that I'm talking about. So I'm going to need someone to keep me on track as far as time is concerned. My wife does it a lot, right? I started my diving career some 50 years ago, right? And where a lot of people go to sea hunt, right? The beginning of my diving career was with a movie called The Incredible Mr. Limpet. Right? Some of the people in the room will already recognize that. Some of the younger people, you'll have to Google it, right? But the idea of the movie was this just gentleman fell into the water and then turned into this cartoon fish. At 10 years old, I'm figuring, okay, right, that's it. Let me get to the beach and I can turn into a fish. That's what I want to do. Waited for my mother to get a family friend to take us to the beach, and I ran and jumped in the water and started to drown because I never learned how to swim. Right? But I figured I would just turn into a fish. Right? Lifeguard saved me, pulled me out, put me on the beach. I said, okay, I must have done something wrong. I waited for him to go away. I turned around, right back into the water, of course, started to drown again, pulled me back out. He says, can you swim? He said, I don't know. He says, well, don't go back in the water until you learn how to swim. I did it one more time before I figured out, okay, I need to learn how to swim. But that was the beginning of my love of the ocean and water. Right? I learned how to swim, and I started diving 40 some odd years ago. Right? In the midst of that, I met this gentleman in the middle of the screen. His name is Kenneth Stewart. Kenneth Stewart was the founder of the organization Diving with a Purpose. Diving with a Purpose started as a result of the far left image is the Guerrero Project. Right? There was a documentary that was being done here in the Keys with archaeologists in the national park. Her name was Brenda Lasendorf. The young lady who created the documentary, Karuna Eberl, was looking for some Black divers to interview while Brenda Lazendorf was the feature of the documentary. So Ken got invited to come down and observe what was going on and said, this sounds interesting, right? So he went back home, right? Called up a number of friends. At the time, Ken was the regional director of the National Association of Black Scuba Divers. Gets another people on the phone, says, instead of just diving for fun, why don't we dive for a purpose and find that slave ship? And Diving with a Purpose was born, right? A couple of years after that, I became a part of Diving with a Purpose and took the course that that they were all learning from. During that same time, and none of this happens in a vacuum, there are a host of other things that are going on at the same time. While I was down here working on the program with Ken, one of the dive instructors for the program asked me to join him with the Coral Restoration Foundation, because at the time, Ken Niedermeyer, the founder of Coral Restoration Foundation, was outplanting corals back out onto the reef. Says, well, come take the class with me. I said, I've done my undergraduate, I did my graduate, I did law school. I don't want to take anybody's class anymore. Right? He says, please, 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 please. I am a wreck diver from the Northeast, New York and New Jersey. We get in the water, we hunt for wrecks. We're traveling the world looking for wrecks. That was my passion. Right? But he kept on pushing, so I finally sat down and took the class. 2 hours, I said, okay, I can give up 2 hours of my life. I sat and found out, oh, okay, those things on the bottom of the water, corals. All right, there's something interesting about that. And then they took us out from the nursery and had us planting corals out onto the reef. Right? So they gave you a scrub brush and a chisel, and like, you're putting me to work, right? I came up from planting my first coral and working in a nursery, a changed person. Right? After having dove for some 25 years at that point, I said, I can do this. Just this, working in the nursery, cleaning fire coral off of these trees for the rest of my life. And it would not be a wasted existence. An opportunity to give something back to the ocean has provided me with so much joy. And I'm slowing down, I'm going too slow. But yada, 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 yada. All right. The image to the far right is a 6-part series on the transatlantic slave trade that was done with Samuel Jackson some 3-4 years ago. I was a part of the documentary with him. He wasn't a part of the documentary with me. Well, he kind of, right? So all of these things are part of what brought DWP kind of to be a common name. During the course of that process, we did a lot of work with the Florida Keys National Marine Sanctuary. And I know I'm going to be, we've already done DWP DWP has an ethos, right? Which is restoring our oceans and preserving our heritage. Most of the work with documenting shipwrecks is restoring our heritage. We're lucky today to have the heritage coordinator for the National Marine Sanctuary sitting here in the audience, Brenda Altmeyer, right? And Brenda is also part of the reason why DWP-CARES collective approach to restoring our ecosystem ended up developing. Because while we were working on documenting of the shipwrecks in the National Marine Sanctuary, Brenda said, Kramer, why don't we look at doing a benthic characterization of the area where the shipwrecks are resting? I said, okay. Then I had to go home and Google benthic characterization, right? Because whenever Brenda says something, you just do it. She just knows everything. She's a staple here in the Keys. So Brenda says it, all right, I'm doing it. Same time, I'm with DWP, we went to St. Croix to do some coral nursery maintenance works with the Nature Conservancy, not Coral Restoration Foundation. And I met a young man named Kemet Amon Lewis. They were building a nursery in St. Croix, right? And after we had done the outplanting of the corals, he pulled out some slates and a transect, and I'm looking like, well, what are you doing? We planted the corals already. He said, oh, well, I'm doing a survey. I'm like, but what? Those are the corals right there. He says, well, there's more than just one coral. There are a host of corals and other things that are attached to the substrate. And I'm like, teach me, Sensei, teach me. So we came up. I'm like, I'm overwhelmed at all of the information. And I know I have an image of Kemet here because he, along with Brenda, right, and a host of other people who are a part of my collective, right, are responsible for the majority of the things that DWP-CARES is now today, right? I can't really take credit for any of it because I have so many people working in and around me that are a phenomenal group. The image, the logo, right, was done by an artist out of California who we went diving. I said, I think I need a logo. He said, well, what are your ideas? And I started spitting out ideas, and he created the logo of the hand in the hand of coral trying to protect the ocean. So I owe that to John Trevino. Right? Advance. Other way. Okay, again, our collective. Brenda, who started, right? Paul Washington, I'm not gonna go through all of these names, right? But Paul is the one who forced me into the Coral Restoration Foundation. One of my partners, Ryan, who built the program with me, Kemet, the young man who did the survey work that got me into doing survey. Once I took his course, right, then I came back and said, there are so many different corals. I created a coral identification program for the NABS members who we were working in the Keys at the time. Right? Ken Stewart, who is the founder of DWP, who frustrates me to no end because he comes up with these ideas over and over and over again. It's like he sits up in the middle of the night and then you get a phone call, 2 o'clock in the morning. Listen, I've had an epiphany. We're going to do this. And you just have to go with Ken because Ken comes up with the ideas and he forces you to make them happen. Dr. Alex Brilski, who's a part of, I don't know if he's still a part of Reef, but one of the most knowledgeable people that I have ever met. Right? And at the time when we met, he's the one who taught me the Reef Check citizen science protocol. And our relationship was such that we'd both been diving around the same time. He has a couple more years on me, right? But Alex was writing his latest book, which is Beneath the Blue Planet, right? Which is a phenomenal book. If you haven't please get the book. It doesn't just talk about the ocean, right? Which amazed me about Alex is Alex, as a marine biologist, started his book talking about the formation of the planet Earth, right? With geology, chemistry, everything. So him, Sam, who gave me money, right? And the people behind the scenes who do all of the technical stuff that I do not have the skill set to do any longer. Who put my photogrammetry stuff together, and I'm going to down. All right. Human impact. When I speak of human impact, I'm talking about everyone collectively, right? What it is that we have been responsible for, right? We're responsible for creating this, right? So we have to be responsible for fixing this, right? We, as a human family, dump over 7 million tons of plastics and garbage in the ocean on an annual basis. It's just not sustainable. Right now, we are pumping 3.6 million pounds of CO2 into atmosphere every second of every day. We're changing the chemistry of the ocean. It's not sustainable. We have to do something to fix this. Hence, our collective approach to restoring our ecosystem. Right? So we work with Coral Restoration Foundation because they are planting corals in the ocean. We work with Moat and the Nature Conservancy. They're planting corals in the ocean. We work with Reef, right? Doing fish counts. Everyone here in the Keys and globally are doing individual things to try and make a difference about what it is that we are doing to impact the health of the ocean negatively. Right? What I have not seen, right, is our collective approach. Everyone is doing something helpful, almost in a silo. We need to be operating as a collective human family to change what it is that we've been doing. And my idea is, one part of it is education. We have to reach into the community, not just scientists and politicians, but us, the community, educating people about the importance of the ocean. And one of the most important things that people fail to realize is that the ocean is where we get the majority of our oxygen. Right? We kill the ocean, we kill ourselves. Right? Just people understanding that as a concept will stop the vast majority of us from pushing our single-use plastics and garbages into the ocean as if it were a garbage can. Right? The other thing is that we need to document the health of the ocean to see where we are currently. Right? We can't fix what it is that's wrong without being able to document where we are. Right? So communication, community education, right? Monitoring the health of the ocean, removing some of those plastics and garbage from the ocean, and the action of restorative healing, right? Trying to outplant corals and sponges and those things that are going to return the ocean to what it once was. The plan of DWP-CARES, which started working in and around the Florida Reef Tract, right, has expanded because we started working with both adults and youth in the National Marine Sanctuary. But some of our kids came from Costa Rica. Some of our kids come from Honduras. Some of our kids come from St. Thomas and St. Croix in Jamaica, right? So we as an organization accept donations and support from a host of other people to bring those kids to the Florida Keys to teach them about how to document the health of the coral ecosystem and how to outplant, and then take that effort and create cohorts throughout the Caribbean, right, to turn the Caribbean into one massive coral nursery. That's the plan. Yep, and I ran ahead of myself. So we started working with the Coral Restoration Foundation some 8 years ago. Okay? And as I indicated, it started with this young man right here, Kemet Amon Lewis. Right? Kemet was a marine ecologist, and I want to say about six years ago, right? While he was also a dancer, professional dancer, he got a pathogen, right? That took made him a quadriplegic, right? He almost died. They flew him here to Miami, saved his life, but he went back to doing work documenting the health of the reef, right? Which is a testament to resilience. So as he was a part of the beginning, I always include him in my presentations. And of course, learning how to document the health of the ocean and being challenged by Alex and what his belief is with respect to our warming oceans, that we may need to pause doing coral outplanting because we've had so many warming events that take place, you end up losing a host of corals here in the Keys. But, right, the argument that we go back and forth with is, what's the alternative, right? We can't give up. And since coral outplanting really started around in earnest around 45 some odd years ago here in the Keys. Right? In 40 years, we've gone from 98% coral loss to 95% coral loss prior to the last bleaching event, which is an indication that we are doing good work, but just that we need to massively upscale the number of people and the amount of work that's being done in order to try and preserve what it is that's going on. And the picture to the far right, is actually how the program ended up including coral outplanting into the collective approach. Because in the beginning, we were just documenting the health of the reef in and around the sanctuary, but we were seeing the reef transitioning from a hard coral ecosystem to a soft coral ecosystem because we were losing so many of our hard corals. So new divers would get in the water and see all of these sea fans and sea whips, the gorgonians just waving in the breeze, thinking, oh, waving in the breeze, waving in the water column, thinking, how beautiful this is. And it is beautiful to see, but the recognition is it's a changing ecosystem. Right? We're moving from hard coral, which protects our coastlines, 360 miles of coastlines from the ravages of storm energy. And I don't have to tell anybody here in Florida about the importance of having a coral reef that diffuses over 90% of all storm energy from coming on shore. Absent those coral reefs, then we get 100% of that energy, which isn't sustainable. So, DWP-CARES collective approach to restoring our ecosystem trains citizen scientists to do coral survey work. We do 4 types of surveys. We do a fish survey, which we're getting ready to change to the reef survey because of loss of fish on the reef. Right? We do a substrate survey. We do an invertebrate survey, and we do a roving coral survey. Our roving coral survey was added a couple of years ago after the introduction of stony coral tissue loss disease, which was killing off better than 20 or more of the reef building corals on the reef. So we were asked by the sanctuary to document the presence of any of these boulder corals, because citizen scientists, divers present a host of different eyes that we can put on the problem and then pass them on to, well, in this area, we would be Andy Bruckner, who's the marine biologist for the National Marine Sanctuary, right, to provide them with the data on the health of the reef. And on top of that, once we do our fish survey, invertebrate survey, substrate survey, and roving coral survey, then we go back and we do a photo mosaic of the area where the documentation has taken place. So now we have a physical, visual manifestation of what the reef looked like at that particular point in time. Right? Right. So, well, that's kind of old, but 30 years of diving brought me to DWP, brought me to Coral Restoration Foundation, the Nature Conservancy. Reef Check was the original monitoring program that we started with, and I developed the CARES protocol, which is a distinctive specialty with Ph.D. now because it includes the importance of hard corals in the data set. Plus human impact equals CARES and you. Right? These are, we have an adult and a youth component. Our youth component, like I said, we take kids from around the country and the Caribbean, Central and South America, right? And train them on documenting the health of the reef. The center picture in the lower portion is actually our team in Honduras this past summer. we were in Guanaja, which is the smaller of the 3 Bay Islands. And we were there to attempt to assess the health of the reef in Honduras when we ran into a group of kids who were doing a beach cleanup. Where am I on time? Doing a beach cleanup, right? Right. And I found out that all of the kids in that community are required to do something, right, to help the island. So in conversation with Green Island Challenge, which is a local environmental organization, so well, why don't we get these kids certified as scuba divers? And then they can document the health of the reef rather than just doing beach cleanup. And then they can collect data on the health of the ecosystem around their island. So that's most of that program. And I believe this lower right is them doing some coral outplanting on Roatan. Right? Far left is one of our newest participants, Cassidy, who was a part of the program. And she was so excited about the program that we require all of our kids to go out and do community presentations. So she grabbed all of the information and created her own presentation, right, and took it back home. And I love when kids are inspired to do extra work, right? And these are the data forms that they use in order to collect the data, and we compile it into one overall report that we submit to NOAA, we submit to Reef Check, we submit to Marine Biodiversity Observation Network, and any other organizations that are interested in the data that we're collecting. And in the lower right-hand portion is last year's youth program. You'll notice that they all have zinc oxide on their faces because people are afraid to put on sunscreen because it makes them look white or palish. Say, embrace it. Embrace the zinc oxide. So we created a ghost diver protocol that says, don't be afraid of saving the ocean. And these are some of the groups of the kids that we work with and certify to work on collective approach to restoring our ecosystem. And not to be undone, the center lower portion is our group of adults that come down and do the same thing. So currently we're working in Barbados doing work. We're working here in the Florida Keys. We're working in Honduras. And next year we'll be in Panama and Curacao. So that's us today. CARES has a distinctive specialty with Patty. We're working with SDI. And now we create distinctive specialties with the other dive certification agencies. We're partnering, recently, over the course of the past couple of months, we're an accredited member with the United Nations Environmental Program. And we're partnering with Northeastern University and the Smithsonian on different research projects. And I hope, well, I don't have to go through the whole bullet, all of the bullets. If you want to support CARES and CARES work to restore our ecosystem or to help youth and create the next generation of marine scientists, please feel free. I am here and open to answering any. Do I need to go back? Yes. And Diving with, I always forget this, so I'm going to say it. Diving with a Purpose is a 501c3 organization. So anyone who comes to work with us on any of our missions or trips can write those things off as a scientific research opportunity. So it's tax deductible. All right. Hey, see, it's there. All right. Thank you, thank you, thank you. Yes, sir.
For the microphone there.
You mentioned Alex Brilski. I took his scientific diver course, 2015, I believe, at the Florida Keys Community College.
Eagle Pro?
Great guy.
Uh-huh.
And one of the things I remember him saying was, whenever you go to the sea, do something for it. Every time you get into it, do something for it. That's probably the best thing that I remember about him.
Hey.
The other thing, I have a question. I'm from a landlocked area up near Pittsburgh, Pennsylvania. And I find a lot of people around me have no idea what's going on in the ocean, have no idea what they're doing and how it affects the ocean. So if I wanted to do a program, say at a library, are there some templates of like a presentation that I could use? Is there anything like this around that maybe Reef has done or have thought about, or any other organizations, like just to kind of give some guidance and some standardization to a type of presentation I could do to educate people in the area?
Yes. Right. I have DWP has members around the country. I am formerly a displaced person from New Jersey. Right. I have done presentations virtually. Right. I've traveled as far back home to Boston to do presentations. I can provide you with access to one of the templates for presentation, right? And we can go through the templates until you're comfortable with the material to be able to present. Yes, absolutely. We have a lot of the organizations, both government and NGO, have moved away from the idea of hoarding information. Right? Most of it is open source, because the whole idea is trying to save the planet, right? Making sure everyone continues to breathe and live, and that we keep the ocean. So holding on to information just doesn't make sense. Okay. Anybody else?
So the corals in
Where am I? Okay,
here.
gotcha. Okay.
Excuse me. The corals in Indonesia, which are very close to the equator, are apparently much more resilient to temperature change than, I guess, perhaps the ones in the Caribbean. Hasn't there been some work about transplanting, bringing some of those corals?
I'm sorry. History is replete, right, with us attempting to correct things that have happened to the ecosystem by introducing foreign objects.
Invasive species.
Right. So even as far as corals are concerned, what we do here, right, is we take corals from here, grow them here, and attempt to repopulate them here. It can have devastating effect to try and move something from one place and put it in the other.
I understand that, but if the corals are not resilient to the temperature change,
scientists have been working on looking at corals that are here, that are more resilient to temperature, and they're working on growing those because they are already here. Right? Yes.
Hi, Kramer in the back.
Okay.
So I wonder if your organization, I love the fact that it's diving with a purpose, has reached out to some organizations that might not dive with a purpose, but that could be involved. Like, and I'm thinking, and maybe incorrectly, because I don't know for sure, but like university clubs, which are oftentimes well-funded by student fees, and any undergraduate student can tell you those fees have gotten very high. Have you worked with university dive clubs to come and at least get an awareness of what you're doing, and maybe bring them to your side of diving with a purpose?
I am working on that. We are working on that. Coming from New Jersey and trying to bring program into the schools in that environment, they were always reluctant to have kids anywhere near the water, right? So my challenge with boards of education is, well, if you're afraid of kids drowning, then you're educators. You're supposed to make them water safe, right? I've also had a challenge getting into colleges and universities, right? But I am open to going anywhere and talking to anyone about saving the coral ecosystem.
There's a number of students in our
anyone.
laboratory at the museum that are now divers and that are very interested in diving. So maybe we can talk afterwards.
Absolutely.
I think that's a great suggestion, Rob. I was wondering if you've communicated with the folks in Key West at the College of the Florida Keys, because I don't know how closely, in fact, I've wondered why Reef isn't more, they don't seem to be active with the college. And I'm an alumni from the early 70s, but the campus here, I don't know if you've noticed, there is a big building up here. It's beautiful. They have a great auditorium and they don't use it. The public doesn't even, seems not to know it's there. And I would encourage you, in fact, I would help you if I can, to make a connection with their marine science program in Key West, which by the way is excellent. They're all set up for studies. They've got the lab. I think a partnership not only with the DWP, but with Reef. There's sometimes politics involved that's beyond the understanding of the general public, but I certainly would be willing to help. And I think universities around the country, if you can find a pathway into their programs, it would be worth looking into. Thank you.
I don't want to speak for Reef because there are enough Reef people here to speak for themselves. We tend to move in the direction that is the path of least resistance. Right? So when you come against people who are saying, oh, that won't work, or oh, get back to us next year, oh, well, maybe if you do this, that, and the other thing, then maybe it'll work. So we're moving and attempting to get things done. Right? So anyone who is open to helping navigate, right, in order so that we can grow, both Reef, DWP, anyone, I don't see where anybody would be opposed to that. Right? I have a challenge with people who have good ideas, but then they put it on my plate, right, to make happen, like because I've got Ken and a list of people who are giving me ideas of things to do, and I don't have the manpower in order to do that as of yet. So we're attempting to grow.
I understand the invasive species issue with respect to the Indonesian coral, but what about, have you investigated or analyzed that coral in comparison to the coral here to see what it is that allows that coral to withstand the temperature changes in Indonesia, and if there's any way to have that impact the coral here?
Those are questions that would be better answered by the marine biologist, right? Not the diver. Right? I have a lot of information, right? But I try my best to stay in my lane when I can.
You mentioned Reef Check. Are they still active here in the Keys, and how would I find them? Because I've been very active with them on the West Coast, but I haven't been able to find Reef Check on.
They are very active on the West Coast. You have my information. In 2 months, I'll be a Reef Check instructor, right? So we can make that happen. Yeah, I've struggled trying to find Reef Check cohorts in this area, and Alex Brilski is the one who actually certified me and helped me move along to becoming a Reef Check instructor. Yes. Ask question in here. Right. I'm answering any questions you want outside as long as I'm here. Yes.
That horrific picture of all the garbage in a bay. 2 questions about that. Was that in the United States or somewhere else? You know?
That's a stock image, right? So I couldn't tell you where that came from. It's just the impact of the image was one that was such that it told a story.
Do you happen to know, like, what entities, what organizations, what are responsible for dumping all that garbage in the ocean? Is it like, if my municipality collects my garbage, do they put it in a landfill or do they just dump some of it in the ocean? Do we know who's doing it?
I cannot answer that question, right? It is both. Can't answer the question. I'm going to answer the question. Both public and private ends up. There are different organizations that own landfills and are responsible for the garbage, and lots of the island countries are attempting to do stuff. Now we've been shipping our garbage to Indonesia. And they just stopped. I can't say they just stopped. It was like 2-3 years ago, they stopped accepting our garbage because they were taking the garbage and sifting through it for some of the recyclables. But they stopped doing that. So we need to stop producing. And right, your question is a good one because we need to look at the people who are responsible and hold their feet to the fire to say we can't continue operating this way. All right.
Thank you very much.
All right.
And as Kramer said, we'll all be out in the lobby in a bit. So if you have more questions or you want to find out some more about the programs that he talked about, you can catch him at the break.
Thank you, family.
Thank you. And I do want to say again, thank you for making the effort. I know with your upcoming surgery, I appreciate that. And Natalia, she kind of alluded to it. She had evacuated from the storm last week, was in Virginia until earlier this week. Ended up getting here by plane instead of by car yesterday. So going above and beyond to help share their knowledge with us as a community. So I appreciate you all making that happen. Okay, so before we break for our happy hour, we wanted to take a couple minutes to, let's see here, do that. Allow some space and, will you pull that up? I can't talk and do that at the same time. So it's up somewhere. Allow us all a little bit of space and time to reflect and remember Paul Heumann. He was a co-founder of Reef. He was a friend. He was a visionary. He changed so many lives around the world through his work with his partner, Ned Deloach, and his collaborations with scientists and taxonomists and divers. And really, it's let's do this. Well, we'll just go here and all right. Well, I guess, yeah, don't start it yet though, because I guess just let's, I don't know, what should we do? Yeah, sure. What happened? You fix it. But it's on like an automated loop, so we're gonna not start the slideshow yet, I guess. You could just hide it. I don't know. It's kind of hard. Okay, we're gonna have Martha start. A collection of us are gonna say some words. We're gonna look at some photos, and yeah. So let's start.
Okay. Well, Paul would be so proud of this afternoon if he was here. The stories that Natalia was telling about jellyfish and how amazing they are, and how that brought together her community and working with her grandfather, to Kramer's work and diving with a purpose, and how we can all make a difference in conservation, and how that collective impact is what's going to change the world. And that's what, at much of what Paul was doing was about community. It was about bringing people together and helping people see the ocean and see the world in a different way. So he would have loved being here for this. Many of you that know me know that I have 2 kids, and while I love being here, there's nowhere else I would rather be right now than spending time with all of you and celebrating the work that we're doing together. But I do miss my kids when I travel. And this morning I was thinking about my daughter. She's 8, and on Mother's Day, she had made a trail of Post-it notes from my bedroom to the kitchen that had cute little sayings. And one of them said, "You are so brave." And I kept that note on my mirror. It's still there at home. And I was thinking about that this morning, and while I wouldn't describe myself as brave, it would not be one of my top 10 words, and I don't think that Paul would either. I don't think that he would have described himself as brave, but he really was. He was a pioneer, he was a visionary. He took risks that moved conservation forward in really meaningful ways. He was a successful lawyer in his younger years, and he left his law practice to start the first liveaboard in the Caribbean. And, while he was doing that, he found that most of the marine life identification resources were written by scientists for scientists, and there wasn't much that was accessible for regular everyday divers. And so he changed that. He worked with Ned to create the first fish ID books and help make understanding marine life really accessible so that everyone can see that they have a place in the ocean. And that took a lot of bravery. Back then, you didn't know how successful those books were going to become and all of the lives that they were going to change. I mean, I had my original copy with the plastic binding, and we replaced the binding. And I know there are people all over the world that I've connected with that have these stories of those books and how it changed the way that they saw the ocean and the way that they understood fish. So the visionary risk that that took is really powerful. The other thing that I think about with Paul is that he had an amazing ability to, if he saw what needed to be done, he would jump in and do it. There's not a resource for recreational divers. Well, let's make one. There's got to be a way that regular divers can contribute to scientific information. Let's start Reef. And here we are all these years later, because of that, let's just do it kind of attitude, to see the way that the organization has grown. Paul, often reflected in the 13 years that I've worked at Reef, he often talked about how proud he was of this organization, of the people that it brought together, the impact that it's had. And he was here at Reef Fest last year, and he got to see the groundbreaking on the new Ocean Exploration Center. And Paul was an early advocate. The board has been talking about potential expansion for more than a decade now. And Paul was always absolutely, we need to do more. We can have a greater impact. And that center is a way to do it. So when we open that next June, I think he'll be there with us in spirit, and so proud to see the work that we've done. So, I think as we're kind of looking at thinking about Paul's legacy and impact and the way that he helped, change the world, really, how he lived a life where he was doing what he was passionate about, and he inspired other people along the way. And if we can do similar things, if we can, dig deep, and even if you're not seeing yourself as brave, to think about what you could do in your community, what you could do in conservation to take that leap and take that risk and move things forward and bring, communities together. I think that's part of the legacy that Paul leaves, is the work that we're all doing together for conservation. So I did make it through that without crying, so that was a big win. And now I'll turn it on to our next, and then we'll do the slides, and then the next person can see if they can make it through also without crying.
Okay, I want to read a few words and then our dear friend Jim Della Pazzi, who was with Paul and I from day one when the idea of Reef was forming in our minds, and also Emily Smith, who is the head of the biology department at Nova University now, was our first intern, among our first interns, along with Christy and Bryce Simmons in 1993, and then followed by Anna. We'll close this out. What wonders have been accomplished by the rare individuals who have mustered the gumption to leave an ordered world in pursuit of a dream. Paul Heumann, a legendary dive pioneer, rests easily within these ranks. Paul, who eventually became my longtime business partner, treasured friend, and mentor, was a promising young lawyer in Wichita, Kansas in 1964 when he took his first fish portraits on a much all too brief dive vacation in the Caribbean. He was immediately hooked and by the wonders and the novelties of it all. However, his hopes of becoming an experienced underwater photographer were thwarted by the constraints of a workaday world. At best, he could only finagle a couple of weeks each year to pursue his budding passion. However, even with limited opportunities to dive, several of his marine life images were published in Skin Diver magazine. Many divers today imagine that the 60s were the good old days of diving. Not quite so. In the early years of scuba, dive boat operators were few and far in between, and access to the offshore reefs were, everywhere were limited. On a spirited adventure one summer, Paul and a couple of dive buddies chartered a cabin cruiser and her captain in Cozumel, outfitted the 30-foot wooden vessel with a compressor, and set their sights south to the unexplored Chinchorros banks off of Belize. The intrepid trio had never experienced such underwater freedom. That early expedition planted the seeds for the exciting idea deep within Paul's fertile imagination. In a bold move in 1974, Paul resigned from his law firm and with a stroke of a pen became owner-operator, mechanic, galley mate, dishwasher, tank jockey, underwater guide, and the evening's entertainment aboard what became the Cayman Diver, Caribbean's first live-aboard dive cruiser. For the next 8 years, Paul and his weekly band of happy guests explored the once inaccessible offshore reefs of the Cayman Islands with abandon. A paramount page in diving's history had forever been turned. To make the most of a good thing, Paul, always a naturalist at heart, without fail, carried a camera every day underwater in an ambitious endeavor to photograph every living species encountered on the reef. This heroic quest was the genesis of what eventually became the most comprehensive library of marine life field guides ever published, and ultimately the foundation of Reef. Thank you.
I'd hope to do this by being able just to say it, but I'm not that good a speaker and it's too important, so I hope you'll indulge me if I look at my notes and maybe even just read it. But I met Paul on the Cayman Diver in November 1979. And for me, that's when it all began. I had been fortunate enough as a young boy to spend many years diving off the coast of my native New Jersey, wreck diving. Sometimes I could see all the way to the other side of my faceplate. I didn't know it during those many years, but I didn't really start diving until 16 years later in November 1979 on board the Cayman Diver, when I met owner and Captain Paul Heumann and began a most remarkable 44 years of friendship. As a young boy, I knew that my future career as an astronaut was over. When my future career as an astronaut was over, I wanted to spend my life on and under the ocean and in the ocean industry, and also to be a lawyer doing it. I knew in my heart it was a pipe dream. A life like that only happened on TV. So it wasn't just that week where Paul served all of us on board. Ned said, as ship captain, dive master, underwater photography teacher, mechanic, entertainer, and storyteller. For me, it was what he began on the last night of that trip when he asked me to help him with the legal advice he needed to sell the boat. For the next 10 years, Paul and I met frequently on Sheridan Street to take care of business and to do some diving in Florida and the Caribbean. But it was 10 years to the week after the Cayman Diver that my life took a second great leap forward. Because in November 1989, 10 years after first meeting him, Paul called me and asked me to make a special visit to Florida to meet his business partner. They needed some legal advice with New World Publications, and they had an idea they wanted to run by me. We met for several days at Paul's house at Sheridan Street, going back and forth on things for New World and things for this new charitable organization Paul and Ned were thinking about. I was pinching myself all along, sure that this couldn't really be happening. Well, it was, and it did, and all of you know very well the subsequent nearly 35-year history of Reef. Ned is very kind to make his comments about my role, but the only thing I'll take credit for is this. We were standing around a tall table in Paul's kitchen one evening, trying to come up with a name. We wanted it to be easy to remember, hopefully even short. But we also wanted it to reflect the coral reefs and the goal of environmental conservation. Short just wasn't possible. We played word games and letter games back and forth all over the place. Finally, Ned threw out Reef Environmental Foundation. Paul said, sounds pretty good. Then, very timidly, I suggested adding education to the name, because that was also one of their goals and was the first word, and the first word would then also be an acronym for the entire name. And that's where we landed. Time for some rum. Some more meetings and some more planning, and 3 months later, I incorporated Reef, and we applied for and received recognition as a tax-exempt public charity. Paul and Ned and all of you took it from there. And because of two remarkable men, their foresight and their unselfish dedication and vision, here all of us and our beloved Reef are today. I say without any hesitancy or fear of exaggeration that Paul and Ned have done more for ocean conservation than any two people in history. Then this second great leap of my life really took off because a short while later, Ned introduced me to Anna. From there, it was full speed ahead. I formed not only an enjoyable working relationship, but a deep friendship with 3 people I love and respect. Although I lived 1,000 miles away, my many week-long trips, sometimes to Florida, formed a major part of my life. Sometime in the 90s, the 4 of us were somewhere in the US, I'm fairly sure it was Florida, at some scuba event, and the 4 of us and a few others posed for a picture. My wonderful wife tells the story of finding that picture, laying on our dresser, and then telling me, "Jim, that is one of the happiest pictures I've ever seen of you. You look like you were on top of the world." I was. Paul, along with Ned and Anna, made my dream come true. I was actually able to have legal clients who were divers and underwater photographers and friends. They asked me to come along, and they asked me for legal advice. I couldn't believe it. These 3 people, so famous and so accomplished, knew my name. They invited me into their homes, took me on the most exotic dive trips imaginable, taught me underwater photography and fish and creatures and corals. And they would have even called me up and asked me to help them and to give them legal advice in their ocean ventures. Pipe dream come true. I'll close, small applause here, by repeating a toast I was privileged to give back in March one night on the Dewi Nusantara in Indonesia after dinner, a place where so many of us have gathered for so many wonderful dives and good times with Paul and Ned and Anna. Many of you are here tonight. A toast I was also privileged to repeat a few months ago at Mango Manor when we remembered Paul. The toast as follows. Paul, we miss you, buddy. You made all our lives a whole lot better.
Thank you, Jim. That was amazing. So now I'm honored to invite up Emily. Emily and I share an amazing start and connection because as was referred to, we both started many years ago, met here, like down the street. It's pretty amazing to kind of help make this journey possible. So she's going to share a couple words. Thanks, Emily.
Thank you. I also wanted to talk from not looking at notes, but I guess years of being a professor. I don't know. I like to have my notes in my hand. But when I stand up here, and I feel very fortunate to be able to do so, I just feel a lot of love. I feel people who are passionate about what they do, want to find more about what they do, want to mentor each other up through the process. Talk loud enough to be heard in the back. So I have been very, very fortunate, and my life forever changed for having known Paul and to become part of the Reef community. I was given a chance, and for that I will be always grateful. It was the early years of Reef. I was 21 years old. I didn't know that I had nothing. But I actually had nowhere to live. I was living in my professor's grandmother's house down the street with Christy, and Bryce was here too. And I just am so honored to be able to say a few words in Paul's honor and really share how deeply I feel part of the Reef family. And although I don't know all of you, I just know because of Paul and Reef, we are all family for that reason. You know, the interest, the love, the passion surrounding the allure of the sea, the joy, the thrill of finding things in the ocean, and this community, it's what brings us together. And our friend Paul, or truly the icon, Paul, who led us into his lives, he was key to that. We can say in large part that all of us are here because of Paul. He was a visionary, he built his path, he started companies. I think he was incredibly gutsy to leave his life as a lawyer, start something that we can all benefit about. He documented taxonomy, he extended the ranges of all kinds of species. He has species named after him. He built his garden into an oasis, which got awards. You know, it got awards, and it's known as Mango Manor. And I think by being able to be part of Paul's world, I was able to grow up and become the person that I am. You know, most importantly, Paul brought people together in his projects and his passions. Reef encompasses all these things. New relationships have been born because of Reef. And I don't know how many families exist today because of Reef, but I know that Christie's does, and I know that mine does. And my husband's in the back making sure that I am trying not to cry. And my daughter, Cynthia, is right next door to him. And maybe many of you have family stories because of Reef. Paul's never, you know, these families have started due to Paul's never-ending love of the ocean and connecting and creating. Paul Heumann made these connections and provided opportunity, and that's what he did for me too, and it impacted me a lot. It changed my life, I'm sure. In the summer of 1993, like I said, I just had been gotten into the University of Miami, and I was funded by the Nature Conservancy at the time, and I was going to work with Reef. So I didn't know what that meant, but I got to meet Ken and Tammy back there, and you know, they made a data sheet. It was paper back then, and we were going to put all the fish, you know, bubble it in on the paper. And so I just come to start with that. And I was so fortunate to be one of the first Reef interns with Christy and Bryce, and Paul provided this opportunity and mentorship. You know, I was able to dive with Paul, but Paul was quite an icon. I could say I dived in the same ocean with Paul. Paul was busy taking pictures of fish, but I was supposed to be making sure we were all writing down fish on our yellow slates. That was before we had paper on the yellow slates. Remember that? We just had yellow slates. And then, you know, I just remember how Paul would smile in a way that would encourage people. And it definitely encouraged me. And he would say, well, Emily, go take care of the data. And I did try to do that. At one time, all the data was in my little rented bedroom in South Miami. And I was really worried that it would maybe stay there forever. But we all pulled together, and with the Reef mentorship, we got it scanned, and it's in the part of the collection now from those early years, even from before 1993 when we had our summer. So Paul made an impact and cared for others. Some of the happiest times in my life were at Paul's Mango Manor, and I will always be deeply, deeply thankful for that and the positive impact Paul has had on my life, especially as I was just trying to find my way and work through my Ph.D. program at the time. I now serve as the department chair and professor at the Department of Biological Sciences at Nova Southeastern University. I'm excited for the bridges that we can continue to build with academic you know, institutions and Reef. So in closing here, to make change takes passion and dedication and really sticking to it. Paul did this, and our worlds are such a better place because of Paul and the legacy that he leaves for us and the challenge that he gives us. Thank you, Paul.
Emily met her husband at Mango Manor, so that was. I have notes too, and I'm going to try not to cry too. So 1 year ago, Paul Human entered this auditorium to a standing ovation. So I think a lot of you were here. And if you were here, you might have heard him in his loud stage whisper say, "Oh, this is for me." And he was really pleased. Illness had prevented him from getting out much the previous year. And if you've ever been on a trip with Paul or attended an event or one of his garden parties at his beloved Mango Manor, or hung out with him here at Reef Fest, you would know how much he enjoyed good company. So he was determined to make it here last year to Reef Fest, especially because it was the 30th anniversary of our volunteer fish survey project. And for months afterward, nearly every time Ned or Eric or Ken or I talked to Paul, and he checked in with all of us once or twice a day sometimes, he marveled over that standing ovation. He did. He was grateful, he was proud, and as he had begun to do over the past few years, he really wanted to reflect on the success of the books and of his pride in Reef. He talked about it a lot this past year. His decision to step back from the board came in early 2020. A major paper about our Grouper Moon project had just been published in a prestigious journal. We had several other big successes, and it just indicated that Reef was shaping up to be, 2020 was going to be a banner year for Reef. And he called early that year to tell me that he felt he could retire because he knew the organization was running so well. So we had a little conversation about that and succession and talking to the board about it. Then he followed up with a long letter, as he could do, about one more thing that he really, really thought we should consider, and that was a new building on the lot adjacent to the existing Reef campus here in Key Largo. So it was something, as Martha had said, that we had talked about, the board and Reef had talked about for a long time, but we just hadn't been able to do it. And so yes, 2020 would be our year to maybe start pursuing that, but we all know what happened in 2020. So we got a little later start. Though Paul passed away before he could see the Ocean Education Center completed, he was able to see the foundation last year of the two-story building as it was under construction. And that Reef Fest was his last public appearance. But he left this world knowing that he'd made a difference. And what more could any of us want?
So thank you all for sharing your parts of the story about Paul. He was, that last year, that was pretty great. We were kind of, he and Troy were coming down, we were kind of delaying, and I'm like, okay, I'll just get started and they'll get here. And I just had stood up to start because we were kicking off the 30th anniversary, and I just started to say his name, and there he came in the building, and it was just like we had planned it, but we hadn't. And he just, everybody, it was such an honor to have this room full of people celebrate him. And I, he knew the impact that he had on all of us. And I know he can hear all of this love now, and knowing that how much he changed us all. So as Emily said, we were young, 21. Bryce was 19. Like, what were we we doing? And down here, we just, Bryce came from Minnesota. I was from Arizona, and Emily came from Maryland. Here we are, and none of us knew each other. And it was an amazing, transformative time for the three of us, and to have an impact, to be able to be a part of something. And here I am. So I never left Reef. I did my PhD that following that summer, and stayed in the loop. And Paul and Ned gave me my job when I finished my PhD, and they could never get rid of me. So here I am, and it's such an honor to be an executive director now of this organization so many years later. And just like everybody said, being around Paul, he inspired us all to do amazing things for the ocean. We are all making a difference every day, and he helped that guiding light of that everybody can make a difference. You don't have to be a scientist. You don't have to be, a taxonomist. We're all that. We all can make a difference, and it's great to be able to see Reef growing. We're going to open the new center next year, and it will be such a celebration to really recognize how far we've come. I will close by, I had an amazing opportunity earlier this year. As someone said, he had a lot of fish named after him. And one of the coolest, I think, is the human's fairy wrasse, human eye. And it's this obscure little fish in a very small little spot in Indonesia. And I had the fortune of leading a group of reef surveyors in April of this year. Many are in the room. And thanks to Anna and her amazing notes, she had a great map, and get in here and swim here, and there's gonna be some pebbles, and you'll wade, and sure enough, we got to see it, and it was really that was just, it was in April, so not just a couple months after Paul passed, and it was a really kind of fitting closure for me with Paul. When I got the news about Paul, I was on the Grouper Moon project, actually, and I know how proud he was of that, and it was great he got to come out and see that project about 10 years ago, and one of those photos was actually from when he came diving. So we could all keep talking, but I think it was a lot. I really appreciate you all allowing us to share this memorial, and we're gonna break, and we'd love to keep talking about Paul and his memory, and then it's 4:45, and so we have about an hour, and then we're gonna come back in here and hear what I think Paul would have really liked: taxonomy and fish stories, and the science of finding new fishes is an amazing, really important one. So let's break, and we'll come back in an hour.